Good afternoon and welcome. There are more seats, like over here there is one. Please take seats. And chairs. While you're preparing, I would like to say a few words before we introduce the speaker of today, welcoming all of you to the first seminar of this year that is organized again by Ariel and Anna, I believe. And I would like to thank you very much for what you are doing. Thank you. And today, I would like to open by reiterating the feeling of mission, enthusiasm, and hopes for the future. We feel like we are in the right time, in the right place. The right time, I mean the right century, the right decade, the right whatever you feel like. I wish I could have what we have here when I was a student at the beginning of my studies. We have the renaissance of brain sciences. And within this renaissance, we here at ELSC and at the Hebrew University in general wish to improve, flourish, and expand the activities in neuroscience. And you can see outside here a new building which will be a brain building, and you see this seminar, and there are many more activities that we will take here at ELSC to promote the brain sciences. In particular, I would like to mention the theory seminars about which you are getting now announcements organized by Jonathan. You're still part of it, and Yoram. Thank you very much for your work. It's highly appreciated. I also would like to mention the postdoc seminars that we start this year. This is taken by Sylvia. Where is Sylvia? Is here, I believe. And she's the organizer of postdoc seminars that will take place also probably on Sundays after the theory seminars. And everyone is invited. We will send messages to this mailing list of the seminar. Anyone who is not on the mailing list of the seminars of ELSC is most invited to enroll and register to be on this mailing list of the seminars. And finally, I would like to mention that we have an open call to all of you for new initiatives, for whatever you think can contribute to our work, to our science, and to our contribution to brain sciences at the university and at the community at large. I would like to give an example just before we start the seminar. This year we will have a one-day seminar for teachers of biology in high schools in collaborations with Belmonte, and some of you, the students, the postdocs, the PhDs, and the faculty expect my request to participate in this one-day lectures and visits and introduction of what can be done in brain sciences to promote the teaching of brain sciences at high schools. And now to the lecture of today. So we are honored and pleased to have Itzhak Fried, an old friend to all of us, and a finalist in this last week competition on the Brain Prize of Israel. Unfortunately, he did not win it like our good friend Chagai, but he made a good show. He had a good show there. So Fried is a professor of neurosurgery and psychiatry, professor of neurosurgery at behavioral sciences of, the Department of Neurosurgery and Psychiatry and Behavioral Sciences at UCLA. He is also a director of Adult Epilepsy Surgery Program. There he also co-director of the Seizures Disorder Center. Now, in addition, Itzhak Fried is an Israeli who is a professor at the Neurosurgery Department in Tel-Aviv University here in Israel. So we are happy to know, I didn't know this, Itzhak, that you studied for undergraduate physics. This is correct, right? So he comes from physics, and this is the appropriate place, the computational neuroscience program here. He started in physics and went to do PhD in UCLA, MD at Stanford. I'm correct. Correct me if I'm wrong. I tried to figure out all the information, which is too long to go on because it will go till 6 o'clock, so I will reduce some of the details. The neurosurgery training you did at Yale University, all in the United States, and he's heading now a cognitive neurophysiology laboratory which is centered on the opportunities to study the human brain afforded by epilepsy surgery. And the epilepsy surgery is one in which he, in small number of patients, can insert electrodes to, you will talk about this too, I'm sure he will tell you why they put these electrodes, but the subject of the talk today will not be about epilepsy, I believe, but about what we can learn from inserting these electrodes in the human brain and studying single unit activity, local field potentials, etc. in these subjects. So the title of the talk is Memories and Volitions, Insight from Single Neuron Recording in the Human Brain, and I hope you will see how important these works are. Itzhak, thank you very much and welcome.
What do you have to do with this?
This in your pocket, this on your side.
Okay. Okay, well, thank you for the opportunity to talk here. It's a real pleasure. It's an enormous audience, really, I must admit, at this hour. I do have a distinct memory, which is talk about memory. A bit, okay. It is a talk about memory. I do have a distinct memory of saying goodbye to Elon in the train station, Lausanne, Switzerland, and I was worried that this may be the last time that I would see him when he embarked on a trip to, I guess, surround the Mont Blanc, right? Okay. And he did it out of his own volition, which was also interesting. In any case, so this is actually a very nice opportunity to talk in length. Last Monday I was really limited to 10 minutes, and now I have a little bit more time, so this is great. So basically, we are talking here about, what I like to call a neurosurgical observatory of the mind, because we do have sometimes distinct opportunity to look at something that can be looked upon in no other environment. And indeed, the process of opening up brain surgery to neuroscience has been a continuous process, but has been recognized more and more recently, with more places really taking advantage of the opportunity to look into some questions which can only be asked in this opportunity. We have, of course, to go back to Penfield in terms of a short sort of historical view, because Penfield over 60 or 70 years ago really was the paramount figure. He started as a football coach at Princeton University, and he went to England on a Rhodes Scholarship and came under the influence of Sherrington at the time. So he was really an example of this interaction. And basically, when he summarized, I often come to this, he had a paper in the National Academy of Science, and he actually summarized his experience in the operating room. And then he said, 26 years ago, I was operating upon a woman, and notice the style of this writing, which is missed in present literature. I was operating upon a woman under local anesthesia in the Royal Victoria Hospital, blah, 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 and she told me suddenly that she seemed to be living over again a previous experience. She seemed to see herself giving birth to her baby girl. Happened years before, and meanwhile, girl had grown up. She was now lying in the operating room, in my operating table, hoping that I could cure her attacks. This, I thought, was a strange moment for her to talk about that previous experience, but then I reflected women were unpredictable. It was never intended that men should understand them completely. Okay, you can imagine reviewer number 2, what he said about that. Reviewer number 3 would have rejected it. Nevertheless, he said, I noticed the fact that it was while my stimulating electrode was applied to the left temporal lobe that this woman had an unrelated and vivid recollection. And first, what was the astounding thing, and you can imagine the excitement in the Montreal Neurological Institute when this happened, because suddenly a whole experience, a memory, a complex mental event was elicited by a crude electrode, which was stimulating a volume of tissue. You can imagine a bipolar stimulation across 10 millimeters of tissue. But yet it was a distinct experience and was always came back with stimulation at the same point. So this is what he called the experiential responses, were always vivid. Two experiences never really activated concurrently, and there was a doubling of consciousness, a beautiful concept, mental diplopia, meaning that many a patient told me that the experience brought back by the electrode is much more real than remembering, actually. But this was a memory, and yet he still is aware of the present situation. It's a doubling of consciousness. So Penfield, after many, if he watched several of those and summarized them, he came up with this statement: there is a permanent record of the stream of consciousness within the brain, hidden in the interpretive areas of the temporal lobe. There is a key mechanism that unlocks the past, and that's as far as he could really get. But this was his, premise at the time. Now, I couldn't, of course, come here and not show this slide, right? So, whether it belongs to my talk or not, I want to show it, and saying that, we forward, here 70 years, and we are in the midst of a human brain project, and then we have, of course, President Obama saying this is the brain initiative, and what is the agenda? The agenda is that we, progressing from C. elegans with 302 neurons, with 7,000 connections, and going to do Drosophila, 135,000, zebrafish, 1 million. The Trojan shrew, which I didn't know, but you can see it on the finger there, which is actually the smallest mammal. The mouse, humans, the shrew actually has only 1 million neurons. So it said we propose to record every action potential from every neuron within a circuit, a task we believe is feasible, and we gradually build up. Now, we cannot really wait, so we are taking the opportunity to look at system and trying to record, from neurons, and not really waiting until we mature from the zebrafish and up, and coming back also to really the counterpart of Penfield conceptually, is really Kahal, and I try to sort of bring this just as a motto in his record of his life. He said, to know the brain, we say to ourselves, is our idealistic enthusiasm is equivalent to discover the material course of thought and will, which I will talk about today. But he said, like the entomologist hunting for the colored butterfly, my attention was driven to the flower garden of gray matter, which contained cells with delicate, elegant forms, the mysterious butterflies of the soul, the beating of whose wings may someday clarify the secret of mental life. So we are looking at those butterflies now in a completely haphazard way, in an opportunity that we have that I will explain. So this is our opportunity, okay? The opportunity, as Elon said, is a patient. And those are patients with severe intractable epilepsy that cannot be cured with medication and have tried multiple medication. So they are candidates for epilepsy surgery. Now, many of those patients, due to modern technology, MRI, PET scanning, video EEG monitoring, we can identify that locus of the seizures and can go in and remove it. But in some, we still have to put intracranial depth electrodes into specific suspected targets. So it's a surgical procedure. The patient wakes up, they go to a ward, and they sit on the ward essentially. They have to sit for a long time because we never know where the seizure is going to occur. So they are monitored 24 hours a day. Usually it takes about 7 to 10 days to get enough seizures so we can conclude whether there is indeed a single resectable focus that we can later go in and cure. And if indeed there is one, the probability of cure can be 80%. So this can be with minimal side effects. It's a whole field of medicine which I don't have time to talk about. But this is really the setup here, okay? So there's a setting. Now, because the patient is sitting awake for 2 weeks, there is really an opportunity to look at the brain signals at various levels, and with the patient essentially doing all kinds of fun things, which I will show you, and most of the patients actually welcome, the break from the boredom of just waiting when the seizure would occur. But our responsibility is really to construct, to ask, the right question, of course, and to conduct a paradigm. We cannot just say, well, let us see, this is interesting, let us try. A project usually will take 3 years because, it has to be built upon several patients, and the technical difficulties are not trivial in this kind of setting. So, how is it really done? There is an electrode that you have the general scheme, of this electrode, which has essentially macro contacts. Do you have a laser here? Okay, there is a laser. You basically has micro contacts here, and macro and micro contacts, which are micro wires, 40 micra in the diameter, which can really give you the multiple neuron and multi-unit and single unit activity. And eventually we have modified it even with this type of microdialysis catheter, which you can have an inflow and outflow of artificial CSF, and through a membrane you can have by diffusion, you can measure the extracellular milieu. So for instance, if you wanted to look at glutamate in the beginning of a seizure, you could do that. Or like we have done, look at dopamine during several tasks, and etc. But it's a very difficult technique. This is extremely challenging. But the bottom line is you can see, like in this kind of electrode, it goes into entorhinal cortex, and you can even match specifically the length of the microwires with the layer, so you can get quite distinct spatial information. So you can, of course, hear those here eventually, those firing, but eventually use standard method, which in this crowd I don't need to explain, to separate, from on a single channels, you can have usually 2 or 3 if you are lucky, and if you are really in gray matter and you are in a certain layer, or you are in the hippocampus, you want to see a field, so you can specifically get this kind of record, which you can use, the several methods to separate the single neuron activity and then correlate it with behavior. Anatomically, of course, we are not at the histology level where we can say, oh, where is our electrode? Let's do histology. There is no histology, but there is MR imaging. There is CT imaging, which shows you, even the microwires here at the edge, and you can superimpose it on the MRI, and you can use various methods of flattening the hippocampus, such as this original paper from Science in 2003, where where Zaine et al. At UCLA have developed this algorithm of flattening the hippocampus, and then putting the electrode in that scheme. You can see, and you can sometimes say whether you are in a particular CA field of the hippocampus, or you are in the dentate gyrus, or in entorhinal cortex. So we can even make, sub-regional statement, although I will talk mainly about the general statement that we have in the medial temporal lobe. You can reconstruct, the hippocampus here, based on the 3 Tesla MR that is being done. So we can have a fairly accurate spatial information. So far about the methodology itself, but here, basically, the system of interest that we have here is the medial temporal lobe. We'll talk about the main structures, which you can see here, and this is the hippocampus itself, which you can see in purple, and then in dark here, in this area here, is the entorhinal cortex with the parahippocampal cortex and the amygdala. In this diagram from Van Essen, the entire visual system, really, it was quite an intelligent diagram, because it did put the hippocampus at the top. People don't realize, but of course, all the information. Now, what is the function of the hippocampus? Essentially, there is one simple fact, and that is every information which you are subjected to now, if you want to consciously remember it later, to consciously remember it tomorrow, and tomorrow, and tomorrow, then it has to be processed through this system. So this is really the role of the hippocampus. Furthermore, every information which goes into the hippocampus really goes through entorhinal cortex. So this is an extreme convergent point, okay? And it is not topologically organized in a columnar way such as cortex is, which present tremendous difficulties, for instance, for fMRI, when you want to look at that type of functional resolution. So basically, what can we do in this type of situation? Well, let's pose some very non-ambitious questions, okay? The first question is, how do objects really become mental objects? This is a question which we asked ourselves. It is phrased in this way. But, if you're talking about Marilyn Monroe versus Marilyn Monroe of the mind, okay, what kind of transformation occurs? Because it's clearly Marilyn Monroe that falls on the retina, or that used to fall on some retinas, it no longer does, or does in a secondary way. How does she become sort of an idea, even a platonic idea in your mind, that you remember, oh, Marilyn Monroe. So how does this happen? First, we cannot trace this process from the retina because we don't have electrodes from the retina at every station of the visual system. But we do have electrodes at the top of this. It's enough. So let's ask and see if we can ask some question. So, and again, just this diagram, perhaps it was not clear, but this is sort of the general flow of information. So what can we do? So we can come to the patient's room, and that's what we have done in the morning, and do an experiment. We will take, essentially, we look at stimuli which are complex faces and complex places and complex objects, and we'll present it, and we'll do the following experiment. So present this series of stimuli, Halle Berry, the Eiffel Tower, Bill Clinton, spider, etc., and neuron number 1. Okay, so this is what neuron number 1 does. Okay. And this is what neuron number 2 does, okay? And we can separate it, as I told you before. And this is what neuron number 3 does. So, of course, we have presented a limited number of stimuli, but we can present 100 and 200. And one observation is that at this high level, we find this distinct responses. There is no response to 80 stimuli, and a response to the 81st. So this in itself was already a surprise. And when you end up with this type of a histogram, and you find out in this kind of screening session, when you present the 100 stimuli, the neuron respond just to 1. Why? We don't know. We don't even have any ambition to really tell. It's too complex of an issue. This is an empirical observation. But then, okay. Essentially, the task is just to say, whether it is a face or an object. Okay, there is just in order so that you will pay attention to it, some monitoring of the attention. But there is no particular task here of memory or anything. Not like you're supposed to encode this. I'm going to test you later about it. Okay, so. But then what we basically come in the afternoon. And essentially, you cannot see it very clearly here, but what we do is we take that particular stimulus in itself, which in this case is Halle Berry, and we just show it in various distinct forms, black and white, color, profile, name written, the name, and we see that those responses essentially persist, and they essentially have the property of being invariant, meaning it is not really dependent on the physical attributes of the stimulus. And at the same time, you keep not having responses to 100 other stimuli. So we're not saying that the neuron only responds to the stimulus. The only observation is that in this spectrum of stimuli, there are such distinct islands, but around every specific response, there is a latitude, essentially, of invariance around it. So there is no sensitivity to the physical feature. She doesn't have to be yellow. She doesn't have to be green. She is that Halle Berry. So that already brings the suspicion that this response may not be essentially subjected to the tyranny of the senses, if you will, completely in this sense. Now, in a later experiment, what has been done here, and by the way, that initial experiment, I should say, because I do have the time to say it, this was done by, it was a paper published in 2005 by Rodrigo Quiroga in collaboration with Christoph Koch, Gabriel Kreiman and others. And here, Rodrigo continued to ask the question, what would happen if I essentially change the mode of stimulation and looked essentially at triple invariance, if you will, essentially a visual presentation of the stimulus, an iconic presentation of the stimulus, meaning the word, and then an auditory representation, in this case like Oprah Winfrey, and he saw this type of responses essentially, meaning that the cell responds also to the sound of the word being actually uttered and to the iconic representation. Now this is what we call triple variance, and indeed, what we found that most of those triple invariance responses were in entorhinal cortex and the hippocampus. They were not in the amygdala, they were not in parahippocampal cortex or in other areas that we recorded for. So this sort of intrigued us because of this cross-modality invariance. Now, what the other thing which was quite strange about these responses, or unusual in the sense that they were relatively late. Those responses actually occur around 400 milliseconds, 300 to 500. We see a lot of those responses. The only difference in the latency itself that we could see was between parahippocampal gyrus, where responses were a bit earlier than that, were like more at 250. But even those are very late. And when you look at the animal literature, and there are not too many non-human primate studies in entorhinal cortex and hippocampus, but in inferotemporal, the latencies usually are 125 or 150. So the question is, what is the reason for this missing 200 milliseconds? Yes?
Do they have to recognize who Halle Berry is? Or did you ever find any responses to someone that you couldn't recognize?
Yes. In fact, the very first, okay. First of all, I should say that we are cheating. How are we cheating? We are biasing, because we essentially come, to a system which has, hundreds of millions of neurons, and we are recording from 80 neurons. Okay, so we are already at a disadvantage. Okay, so we do choose stimuli from the milieu which is known to the patient. Our rationale eventually is that this is a milieu where they create a lot of association, and we bias and increase our chances of fishing this type of neurons. Okay, by the fact that we say, okay, what do you like? I like actors, I like, race car drivers, I like sport, I like these kind of things. So we essentially bias, our now, but in the very first response that I ever saw, was I presented the Ekman faces. I don't know if you know the Ekman faces. And there is a guy there called Bob. He's not particularly handsome, and there was this. The patient had a response to Bob. The patient didn't recognize Bob, didn't know. But out of all the Ekman faces, there was a huge response to Bob. They're all black and white, and no response to the other stimuli. So there was a coding. Now you would ask, why did they respond to it? And you can say, well, maybe he reminded me of something. Maybe there was some association associated with it. But these do not have to be necessarily stimuli which are familiar to the patient.
One question, is this a restricted small spot? Or
I'm sorry, what?
Is the response restricted to one local spot, microspot, or can you find the same response several places?
Okay, again, if you're talking about a specific site, you're talking about in the hippocampus itself. Yes. Is there neighboring cells which have the same type of response? Usually not. That is, you can find cells which are very close by which respond to totally different stimuli. But you also can find several cells which respond uniquely to the same stimuli, or you can find a cell which out of 100 will not respond necessarily just to one, but may respond to 2 or 3. And in fact, when you look at it, as you go up the hierarchy and go to the entorhinal hippocampus, you find responses to a smaller number of stimuli out of a choice, let's say, of 100. Okay, This, this goes into a long discussion. Maybe, I'll try and talk about it a little bit more, then you can ask this kind of question. But in this experiment, which was again done by Kirog and Mukamel, and together with Rafi Malak at the Weizmann, basically we presented stimuli for, with a backward masking paradigm for 33 milliseconds and up to 264, essentially testing the recognition of the patient and looking at the responses of stimuli which were recognized versus those which were not recognized. And essentially, you can see that when the stimulus is not recognized, which is red here, there's really no response. So when you compare actual recognition to no recognition, meaning they couldn't tell anything about it, essentially, then the response is missing, meaning that somehow it is connected with conscious perception. And we have several studies, which show the same type of phenomena. We now have done it essentially in a binocular rivalry situation, where we really show that you really have, that only when you perceive the stimulus, it's not the retinal image, it's really the conscious perception of the stimulus which elicits the response itself. We have a flash suppression paradigm that we did already 8 years ago by the work of Kreiman together with Koch, but now we actually have binocular rivalry, which is a very difficult paradigm to run at the bedside, but even then, we can see this type of connection. Now, how are these responses formed, okay? And here, what we have seen it, that we had an opportunity sort of to look at, and we included in our stimuli, essentially the images of the experimenter. So what happens is in day number 1, the experiment comes in and say, hello, my name is such and such, and they started running the experiment, and there is no responses, okay? In day number 2 or number 3, you can start finding this type of invariant responses, essentially. So you cannot see it very clearly here, but this is one of the experiments, one of the first author of a Nature paper, and he basically you find responses to this person, Arna Ekstrom, okay, that are formed later. So these are dynamic, and they are probably formed all the time. These are not necessarily static responses. How they are formed, we don't know, but they can be missing in one day and then acquired along several days. And so looking at this issue again, this is again a complex paradigm, but it is in preparation. The paper is not published yet by Matthias Eisson. And what Matthias did here, essentially, he took essentially places and faces initially. And so initially there is, let's say, a response to the White House, and there is no response to this character. Now you present a situation where the patient is meeting that person in the White House. And after, a few trials, you basically get an established response to the person themselves, even with no task, essentially. Just by presenting this association, where the task is not to remember necessarily who you have met. When you present it as a pair, for several times, then this kind of response is acquired. A cell which did not respond to a particular person starts responding to that person, the cell that has responded to the location, essentially. So again, this all, essentially, we believe that they are formed by association. That is why when we are essentially presenting to the patient familiar pictures or pictures from areas of interest, then we believe that we are tapping into a higher or more richly associated field, and we bias our chances of essentially getting a response. Okay? So, we essentially have looked at this and would essentially say, looking, for instance, at Einstein right now, the bottom line is that it probably takes you 200 milliseconds, probably less than the latency of these responses, to recognize that you are dealing essentially with with something which is similar and has some kind of representation in your brain, regardless of the color of the bicycle, of the mustache, or the hair, essentially. So the code is an interesting code. It is specific yet invariant. Okay, it is late, meaning 300 to 500 milliseconds. It is a conscious code, meaning the responses are present only to consciously perceived information. It's probably associative, meaning that it can be transferred to a stimulus which is associated to S2. When it's associated with S1, when the response was just to S1, it is sparse, and again, we can talk, for a long time about the numerology of this whole thing, and what is the probability of finding this, and how many cells can represent essentially a concept. And there is a paper that we published on this by Wade together with Christoph about that sparse representation, meaning, okay, how many neurons do you need in the hippocampus to essentially represent a concept? And using a model which is fairly conservative, we ended up with 1 million to 2 million, which we regard as an extremely small number, or at least very different from the idea of a completely distributed code. And it's probably, I suspect, it is even less than that. So it's a very efficient and sparse code, and it is abstract in the sense that it is not sensitive to the physical features of the stimulus, at least not at the way it is done at the lower levels. Yes.
Do you have an estimate how many different concepts a single cell is involved in?
How many? Okay. Well, again, I can only say, okay, how many concepts there are, do you think? Do you have an estimate? Well, 5 is probably not the answer. 50 is not the answer. 500 is not the answer. 5,000, probably not. 50,000, maybe. Okay? So intuitively, but there are psychologists who, look at this kind of question. Okay, we're having 100, okay, and then we find in the hippocampus, on the average, the cell will respond to 1.7, and in parahippocampus, it may respond to 3.5, or you're presenting something from a similar category, outdoor scenes, you will find out that the hippocampus, yes, cells do respond more readily to outdoor scenes, okay? And if you record it, from the fusiform face area, as Doris Tsao has done, it's probably you drop an electrode, you will find you have a very high probability. But this is what you find in the hippocampus, so 1.7 out of 100, let's say. So do your math here. Now we are biasing this because we're not talking to them about penguins in the North Pole. We're talking about Jennifer Aniston, who is the next door neighbor in terms of reading, or in terms of the magazine that they're seeing, etc. So obviously it's a great area for computational neuroscience. Okay, so maybe we can speculate at least about this. Okay, so from this, we came to this issue of these are essentially concept cells, and they're important essentially in the transformation of information, probably from percepts into something that you will consciously remember later. Now, tomorrow you'll remember that I gave a talk here. You will most likely have this memory. You may not remember, that I wore a white shirt, or you may not remember the details of my appearance here. But of course, this is useful information. So obviously, the hippocampus is not just recording, concepts. It's recording specific information. Indeed, there is this whole issue of to what extent pattern separation is represented in the hippocampus. There is an experiment which we haven't published by Susanna et al. At UCLA, where here actually the task was, to take this Meg Ryan, this specific representation of Meg Ryan, and then essentially presenting later a choice of recognition of the specific one that you have seen, where essentially there are lures, and the lures may be Meg Ryan a little bit different, or you can have a totally different actor, which looks like her, but there is a grade here of similarity, and you do find, as you might expect, you find several types of cells. If you look essentially at Shrek here, Shrek gets here an invariant responses. Essentially, every Shrek gets a response, and somebody who may be a little similar to Shrek but not very, doesn't really get a response. But this specific representation, and you can see the same thing that is happening in Bill Clinton. But at the same time, you may have responses, which are present to particular individual. So here, for instance, you can see a very strong response to one representation here. So also, and this actually found mostly in CA3 and the dentate gyrus, and it really goes along with what is thought in the animal literature. Now I'll take a digression, essentially not a digression, really to complete the picture, and you can see that complex circuitry where the entorhinal cortex is essentially at the entry to this system has obviously been studied in a spectacular manner in the rodent literature. And in a sense, there is really a gap between the rodent literature and the non-human primate literature and the human literature. But just to refresh your memory, I mean, the main types of cells that we see are essentially the place cells, which are thought maybe as combination of multiple grid cell representation. So the grid cells and the place cells have really been at the heart of this entire complex circuitry in the rodents. So we basically asked ourselves, years ago, we said, well, why don't we do that? And for a while I said, it's really a boring question. We're not interested in replicating the rodent stuff. But eventually we ended up doing this study. It's not we don't take the patient and walk them, in a maze, and we certainly don't put them in a water maze. But we basically present them with a virtual environment that they can navigate in. And we have this, taxi cab task, yellow cab, where they have to navigate a city with X number of stores, and they take passengers to stores, and that's how they learn the layout of this. So when we use it already in 203, and this is how the task works. It's not completely immersive. We are working on virtual reality to make it more immersive. That's probably why, the results are not as spectacular as the rodents. But this is what the patient is essentially doing. They essentially shuttle passenger to stores, and then they are given the next store as they are moving from store to store. In this environment, we do see that particular spots in the environment, you do get cells which increase their firing rate. But I tell you, it's not as clean as the results of the rodents, although we can see that type of representation. In the same way, in this open environment task here, we just published essentially grid-like, just last month in Nature Neuroscience, the grid-like representation with the six-fold symmetry that you essentially find in the rodents with the same criteria that they use to identify grid cells. We see those here, and I've just seen, an interview with Moser the other day. He says, well, I'm relieved that we, found the grid cells in humans now, but of course they're not as clean, as the rodents. And of course, we are not rodents, and we're also measuring this in a noisy, UCLA ward. We're looking at one neuron in that system. But even with all those limitations, we can really see this six-fold symmetry. And even, nicer to some extent, some observations which I even like better, is that when we look we find directional path cells. So when we put a patient in an environment, essentially moving clockwise or counterclockwise in this circular maze, you will find cells which fire when you go in a certain direction, like counterclockwise in the maze, and they're completely silent when you walk in the other direction. You can see it here, essentially in this cell. This is a paper by Josh Jacobs with Mike Kahana. I have to press it again to see that. So you can watch the trajectory here, but so here they're moving in one direction in the maze. Okay. Okay. So don't underestimate this observation. It's a single neuron. It's a noisy ward, We're fishing this out, in this type of situation, which is not even completely immersive. But when they move in the other direction, the cell is really silent, essentially. So, what is also nice that we find, we find unidirectional place cells, meaning that in this circular maze, there are cells which fire at a particular spot, but when you move only in one direction, when you move back, you really do not get this. So obviously, when you navigate, a spatial environment, these are only a small subclass of cells. Obviously, you have to take head direction into consideration. You had to take many other variables and the visual stimuli. I think for us, navigating in an environment and negotiating an environment with all the visual stimuli, the representation is obviously more complex than in the rodent, very schematic maze that they have to negotiate in those experiments. And as a digression, I would just point out that, so we asked ourselves, really, can we intervene in this system in some way, indeed? And what we essentially did here, we took that same yellow cab task and we applied, actually, stimulation in entorhinal cortex and in the hippocampus itself while the patient was learning the maze itself. This is a paper from the New England that was published last year. Again, the first author was Nancy Susanna. It was in collaboration with Barbara Norton at UCLA. But here, essentially, the patient is again in this virtual city. There is a passenger coming and says, okay, take me to this store. And what they can do is, let's say this is your target store, they can go directly toward the store, or they can really walk all the way around and eventually get there. So you have a variable which measures their performance in the maze. It's called excess pass, the extra mileage which you essentially waste, as you go toward the target. The battery works. We gotta keep on talking as you do this now. Scream so you can hear me in the back. So this is a variable which measures their performance, and what we do here, essentially, we have 3 blocks where we stimulate in half of the trials and do not stimulate in the other half. And in block number 4, we basically test the performance. Okay. And so what you see here is quite interesting. In the hippocampus, when you stimulate, essentially there is no difference between the performance with stimulation or not, meaning those stores that you navigated to with stimulation and those stores that were not stimulated during the learning period, there is no difference. But in entorhinal cortex, there is a difference. When you look at the performance of the memory for those trials where you were stimulated on, you remember those better, meaning that your measure of extra mileage or the measure of the birbur and the excess path length is really longer without stimulation and shorter with. And again, what we think essentially is that it's an issue of the theta rhythm, because when we measure the theta rhythm in the hippocampus itself, when we stimulate at entorhinal cortex, we really see that there is a resetting of the theta in the hippocampus with stimulation in the entorhinal. So I'm just mentioning it's just a digression, but a real question is really how does semantic memory and episodic memory relate really to the whole literature of place cells and grid cells and spatial navigation. There's an interesting article, just appeared in Nature Neuroscience where Buzaki and Moser were trying to speculate about this and essentially comparing both episodic memory and semantic memory, to the representation which you have in the rodent. So in a way, I would look at the concept cell that we are describing perhaps as a form of semantic memory. Now, what happens about episodic memory, essentially? What can we say about that? Well, first of all, the representation that I've showed you is strikingly sparse. It's not a distributed representation, and always reminded me, in a sense, of the sparse representation of the place field. It is the unique ability of the hippocampus to record a measure of individuality, essentially, which is surrounded by invariance, okay? But at some point, we wanted to move to episodes, meaning we wanted to present the patient with an actual episode, not just a static stimulus. And that was a very ambitious study because we wanted to present the patient with a string of, let's say, videos which represent episodes, and then come and do the most simple experiment that you can think about, just ask them to recall what they have seen. And when they recall, there is no physical stimulus. There is nothing. Basically, you are locked in your mind, and things come back to your mind somehow, and they surface on the platform of consciousness, where you suddenly say, yes, okay, the memory comes in. We were interested in that process. Now, unfortunately, it's an n=1 experiment, meaning you only have one chance. You get, okay, remember now, yes, I remember. Remember again, I remember. No, you just get one chance. And you are facing a single neuron, and you are on the UCLA, what I told you, it's noisy, it's terrible, and you are facing a selection, and all the numbers are against you. So no sane graduate student will take such a project, but indeed we found somebody who undertook this, Hagar Yelbasagiv at the Weizmann Institute. They support students for many years. I said, okay, let's do this. And it was done with Rafi. Essentially, you've probably seen this, but again, what happened here, first what we have seen is that there were essentially, and that's essentially how does the object or the episode is selected for conscious representation. Let me just gloss over that and told you the experimental paradigm. You have a viewing session where you review a bunch of clips, and then you have a distraction task, which will appear here, and then you essentially have the recall task. That's nothing on the screen, you just remember, okay? So first of all, what happens during the viewing session here? In this kind of example, you can see the videos being presented. Okay, I guess we didn't see that. Let's see, maybe I'll convince that clip to work. Yeah, it will work eventually. No? This will work? Okay. So this is how you
moan and the miserable groan from the pain that you felt when I was alone. What you doing, sucker?
Everything in terms of all the isms that we live with, racism, sexism, homophobia, the whole business of being human.
There are many in between. I'm just showing you a few where the cell is very silent, okay? But when that laser comes back,
everything in terms of all the isms that we live with, racism, sexism, homophobia, the whole business of being human.
How reliable this response is, okay? And how silent the cell is to many other stimuli, okay? You can say, well, it's an uncontrolled experiment. I mean, I want to know exactly all the colors. I want to go from the retina. I want to understand
everything in terms of all the isms that we live with, racism, sexism,
which cannot be argued.
The whole business
for some reason. Okay? So what you get is something like this. You got all these various videos where the cell is completely silent, and there is one where it's completely out of control. Okay? Why? We don't know. Okay.
Welcome to Hollywood. Everybody comes to Hollywood, got a dream. What's your dream?
I have a dream that one day.
Welcome to Wall Street and the New York Stock Exchange, the world's largest and surprisingly one of.
Hey, Dad! Right on time.
We're getting out of here. Now, jump in,
there's fire.
Mark.
I cannot hear it for some reason. Okay, fine. Okay. But the more
Welcome to Hollywood. Everybody comes to Hollywood, got a dream.
Video. Yeah, we don't have the. Okay, no. This is okay. This just shows you that. But essentially, the other video. Okay, so here the patient is recalling what she has actually seen. Okay. Remember something about New York that was there. Okay.
Hollywood sign.
cell is you cannot hear the firing, unfortunately, because for some reason only one channel comes through this tape. But obviously the cell starts firing. Essentially, what happens is that 1.2 seconds prior to, the cell actually starts firing. And what you get essentially in entorhinal hippocampus, you get firing during viewing, and you get this type of response prior to the report of the memory. And you get the same type of viewing responses in other area, frontal cortex, amygdala, hippocampus, but you don't really get the type of recall. So believe that this is indeed now listen, look to what happened here. Here was a cell that was essentially responsive to the Simpsons, essentially, and the patient was trying to recall. Now she's going to tell you, okay, now I can only tell you that the cell did not respond to Bill Clinton, because this was, one of the stimuli that we basically used, okay? So look what happens here. This kind of thing that if you listen to what the patient is saying and you are alert enough, then you will actually have a rare insight, perhaps, to the mechanism. Okay. And I was surprised. She says, cells start firing. Bill Clinton. Why? Now she explains to you why. So that's it. I mean, completely spurious, okay? Completely spurious. So this is what I'm saying, and of course, we are only in the beginning of trying to understand it, but it's probably the association here that was spread that created it. So what is happening is then our model is such that indeed you do have, and when you summarize the response, you do have an increase in firing rate, and at some point you're basically saying, yes, I remember, okay? Do you have about 10 minutes or what? What do I have? No. 5 minutes, okay. Well, so we will move through, fascinating. Now, I won't even show you this because you'll just be mad at me that I am not showing you this later. I will just point that, to the other part that I wanted to discuss, of course, and I never reached, is essentially, I wanted to go very briefly and discuss this, and essentially the idea that in a totally different set of observation, when we stimulated in the supplementary motor area, we found out that the patient did not have a memory. The patient essentially had a feeling of urge to do something by stimulation. The same way that Penfield has aroused the memory, we have aroused the volition in the medial frontal lobe. Okay, and we have studied this. We took it, a long way, and the bottom line is that we use that Libet paradigm that you all know about, and in the Libet paradigm, to those of you who do not know, Libet essentially presented his patient with a clock. He said, when you feel like, just press a button. And then, but watch that clock, essentially. And the patient pressed, and notice when you feel the urge to actually do it. And the patient actually timed the time on the clock. They said, aha, this happened, my urge happened at that time, and then the position, the pressing of the button happened, let's say, 300 milliseconds later. And of course, he basically, described what has been known before, the readiness potential. But what he showed is that readiness potential prior to movement was already present before the patient consciously recognized that they wanted to do something. So we have taken essentially the same, paradigm and put it essentially in single neuron recording from the SMA. And what we have seen essentially is this type of responses that I'm describing to you here, that prior to that point where the urge arises, there is an increase in the firing rate of supplementary motor area, so that suddenly there is an urge. But of course, this urge can be predicted, and we show that it can be predicted before you recognize that you have an urge. in the same way that we can predict your recall before you recognize that you have a recollection. So essentially, and you can see actually here in this situation what happens, how the cells actually increasing the firing rate, essentially. The firing rate increases, and the patient says, I want it, okay? But the firing rate it is, of course, prior to that event in itself. So that the model essentially, if I will go right forward, is essentially that you do have, both in the medial frontal lobe and in the medial temporal lobe, you have two parallel processes. Now, we're all very upset when we are told that we do not have free will, but we're not very upset when we're told that our free recall doesn't work, right? Connectivity to age, oh yes, I just don't remember, so what? But why I don't have free will? That's upsetting. But essentially, these are the same mechanism, meaning, essentially, that in the medial temporal lobe, you do have an increase, or you at least can observe an increase in firing rate, and at some point the patient says, I remember. You can predict it. In the medial frontal lobe, you do have an increase, essentially, in firing rate. And the patient at some point tells you, oh yeah, I have an urge, but you can predict it. And we show it in that neuron paper from 2011 with Gabriel Kreiman and Roy Mukamel from Tel-Aviv University. We show, indeed, that you can predict 500 milliseconds prior to onset of the volition. With 70% accuracy, you can predict the fact that you will have that type of urge. So in a sense, this is a juxtaposition of memories and volition, and the origin of the title of this talk. Obviously, we have erred on and biased the talk toward memories, but please let me know if you have any questions. Thank you very much.
So it's like a third person observing from the outside.
Yes.
So what is this? I mean, it's a behavioral paradigm that you are talking about yourself in the same way that you're talking about others?
In what way?
Well, if I have to observe myself first in order to know that I have an urge, so it's like I'm observing you that you have an urge.
So what are you saying? The paradigm is not a valid paradigm of will, or what?
Right.
Yeah. This is one of the criticisms that has been leveled against the Libet, the Libet experiment. But the effects are clear, essentially. The observation is clear. Okay, and when you do it under this condition and be replicated times and again from the phenomenological standpoint of the experiment, Patrick Haggard, did that over and over again. The observation is clear. The interpretation is completely open. Absolutely agree with it. So, I mean, we have done it, in a much more ecological way, in the sense that we have put a patient in a car, and the Libet clock was at the dashboard. Okay, and now you just had to make a decision whether you are turning right or left. Okay, but we could predict the decision before you made it. But one criticism was that the process of self-observation is not valid. Okay, or has some flaws. But this is the best we can do.
So who is observing the inside the skin and outside the skin? Who is the observer?
The observer is the patient. The observer is the patient. Can you take this? Oh, okay. Yeah. Fine, I'll do it. Sorry. Yeah. You're talking about the temporal window?
The person did the experiment several times. Except they have an urge. You can show that the neurons are firing 200 milliseconds beforehand. Given the time of the previous trial, can you predict when that will happen, when that rise will happen?
Yes, we can. And again, I will refer you to the paper itself in Neuron. So we are not dealing just for prediction if he will indeed do it or not, but on the timing of that. And yes, and you will read the paper itself. With a certain window of time, you can predict the timing also, the timing of the will. Yeah. Yes.
So how valuable is that time? If human beings do not inbreed, they're much less accurate than mouse in the lab.
You are talking about the time prior to will. I mean, there are fMRI studies which state, and I don't know if I believe this, that this is like 10 seconds, but this is hallucinatory, because they are looking actually, and they claim that this is actually when they record at the medial frontal but very anteriorly, they can get a signal in the fMRI with all the limitation, 7 seconds or 8 seconds. What we are seeing is the same time window that Libet has seen, recently, from 500, I think, to 1500. This is sort of the window that we see that.
But those experimental paradigms are different.
Huh?
The two experiments, this is Libet-like experiment.
This is a Libet-like experiment, yeah.
The other ones are
the
the paradigm itself is from
yeah.
one
Yes.
because the ball is slow. So
Exactly. So there is a limitation of the bold. The prediction there is 56. So it's not an astounding result. Yeah.
What happens if you have to change your mind by doing something else, slightly by a quick flash or something?
Okay. Libet himself was very upset initially because of this, and he has to reconcile it with him being a good, Christian or whatever. I don't know what he was. And he said basically, we do have free will, because what happened is we can, in that 300 millisecond between W, will, and P, performance, we can veto the decision. We can change our mind, and we can say veto, known as the veto hypothesis or the free won't. So we do have a free won't in a sense. This was his interpretation, yeah. But we haven't tested. I think, Haggard is doing now experiments where he's encouraging the subject to change their mind, exactly. But we haven't done it. Somebody who didn't ask yet? Yeah, okay. You go ahead. You choose it in
I have two questions, but I'll start with one. So going back to the early experiment that you showed at the beginning, I noticed that you had a picture of Darth Vader and a picture of Yoda. So did you ever see a cell that responds to a more complicated, abstract concept like any character from Star Wars?
Yes, and it happened to us in an embarrassing way. Because when we submitted the 205 paper to Nature, and after it was published, somebody wrote views and news about it. And it says, did you notice in your supplementary figure number 92, yes, that actually the cell didn't just respond to Jennifer Aniston, but also to Lisa Kudrow. I said first, who is Lisa Kudrow? And then after that, but you're absolutely right. We see this type of thing. And again, this is the association field, which is, you say maybe not, maybe yes, but this is what we occasionally see. Yeah, but how does it come to represent a combined concept? That's the question. Who is next?
Can you rewrite memory, like in a way, like, if you approach the cells that encode like the symptom, and you try, if you can approach it, probably you can stimulate it. So maybe you can do some stimulation that actually releases the memory, and then it will be very interesting to see if you can confuse the memory and show it like pictures that are like
that are similar, yeah. Obviously, those things can be done. But I tell you about stimulation, it's much more complicated. I think that specifically to stimulate and to eliminate a memory, obviously eliminating one cell will not eliminate the memory. And because it's not, I don't believe it is topographically organized, I think it will be difficult. And furthermore, micro-stimulation will be extremely difficult in this type of setting. We are struggling with this issue now. The stimulation which I've presented, by the way, is not micro, it's macro. So if we're stimulating something that is still very low in neurosurgery, 0.5 to 1 milliampere, but this is macro stimulation of the entorhinal.
So the big picture of the first part of the concept cells. So would you say that in the human brain, for every concept, there is a small group of cells that aspire for this concept, like that representation, or?
Yeah, I think at this level here, yeah, I can only talk about this system here. Yeah, the hippocampal system. Yeah.
So this is
yeah.
Every word in the language will have a group of cells that fire, and every concept will have. So are there enough cells?
That's a good question. Are there enough cells? Well, obviously, those groups are overlapping. And I wouldn't necessarily, I wouldn't think that those are distinct groups that are islands. In short, no representation is an island, to quote a famous poet.
But that's the picture.
That is the picture. Yeah. And again, I think it's a great question for computational neuroscience to really, are we running out of neurons? Okay, because this is obviously a system which is not that great. I mean, you can say, well, it has several hundred millions of neurons. This hippocampal system is a relatively small system.
It's not only that. I would like to add a follow-up question. The question is that, say, this week we watched several methods to look at neuronal activity. Brain-machine interfaces can only work if you have a population of cells and you give different weights to different neurons to decide about a concept. In that case, movement or something else. Okay. So you completely neglected this in your approach, I think, in all the studies. You really neglect the issue of that the population is much more efficient in number of cells than single neurons. And I believe that the problem is that the stimuli, the coding is sparse because the stimuli are so sparse themselves. You choose some stimuli from a huge variety. But if you had some continuous measure of consciousness or of memories or something, then maybe I would like to know what you think about how to approach the population level, which is really seems to be the way the brain must work, right? You agree that even if it's groups that overlap, you go into the issue of populations, right?
Yes, absolutely. And again, there are two ways about it. One is that you can record from 1,000 neurons, at the same time. The second, that you can look at the local fields. Essentially, this is really your only options. Really, your only options really is to look at those two levels with current methods. I mean, do you see any other one?
Well, if you can put multi-electrodes in there, it will record for you 100 cells.
Exactly.
To the other.
Furthermore, if you record it from different areas, of course, obviously, this is what we can do. With this technology, but I think it's your statement that it's no wonder that just one cell responds to one thing, and it's not at all trivial. In the same way that you could say that the cell, that the spatial representation will not be essentially by place cell, but by some kind of combination of the entire neural system in that area. So it's still, I don't think it's a trivial observation. Now, about how efficient a brain-machine interface, obviously even brain-machine interfaces that are based on 100 neurons may not be that great. And the question is, do you need to combine local fields with? I mean, you are the expert in this. But if I had time, I can show you that brain-machine interface composed of 2 neurons, essentially, or 4 neurons to be accurate, in which you can essentially change the image on the screen, based on your volitional control, in a system which will be very reliable as long as you identify the right neurons, of course, which make it impractical in that way, right?
I have a question about the interpretation of this being will. And we know in psychophysics that behavior in consecutive trials is almost always correlated in some way. So one can, to some extent, predict what the person will do in the next trial based on what the person did in the previous trial. If this is true in some sense, this can be thought of as memory of the history rather than predicting the future. In that sense, I guess that's interesting.
Okay, it's an interesting interpretation, but the observation is still an observation. That you do basically, I mean, you are arguing about our ability to predict, but you're not arguing about the effect that a cell will increase its firing rate prior to essentially a movement, or prior to self-observation, as limited as it is, of what one feels. Okay?
But it's a testable question whether the cell activity can predict something beyond what you can predict based on the past.
Yes, absolutely. No, this is good.
That's what we statement about the cell. I mean, you are using a classifier or predictor based on the cell activity. You don't take into account the past. Whether the cell is okay. So the neural activity now has information about what happened. I don't know,
about the future.
Because that's what you're using to use code.
But this could be due to the dependence on the past.
The cell may use it, that's fine. Yes. Of course, the cell is predicting by some...
How can you predict the future without knowing the past? This is how you predict the future. You learn the past and you predict the future, right?
That's the question about the mechanism of the neural responses. But the neural responses have information about the future.
Okay, it's a question about information about the future. I think there's a meaningful question here that you can ask about the statistics of the firing relative to the decision.
No prediction about the future can be done without correlation.
Without information, you don't know where the future is coming from.
That's the most enjoyable part of a talk, watching the audience argue among themselves.